bacteria to make some protein for us, a specific protein that we gave them the instructions for. But we don't want them to just make a little bit. We want them to make a lot. And so when we talk about making it, we call that expression. And so we can refer to overexpression when we get bacteria to make a lot. And we're going to make them make so much that they're going to actually shut down basically everything else they're doing and just focus on making our protein, which is great because we'll get lots of protein. But not so great if you basically don't allow the bacteria to grow to have enough of the bacteria to actually make enough of your protein. So the trick is you want the bacteria to grow without making your protein, to grow to high levels, but not high enough that they're going to start like dying off, but high levels. And then you make it so that you tell them, okay, now start making our protein. And we call this induction, when we kind of induce them, get them to make our protein. And the way that we're going to do this is by taking advantage of the regulatory machinery that the bacteria use in order to regulate the production of the enzymes that they need to break down the sugar lactose. And so these bacteria really like glucose, and lactose is like a backup, because from lactose they can actually get glucose. So the sugar glucose is a component of lactose, but in lactose you have that glucose bound to galactose, so another sugar. And so in order to free up the glucose that they really want, the bacteria actually have to break down the lactose. And so they have to make an enzyme, β-galactosidase, in order to break that up and get that glucose. And so if you give them glucose already, then they're not going to bother breaking down the lactose, even if there's lactose around. It's kind of like, why break open a peanut shell if you're given the unshelled peanuts already? And so they're not even going to bring out their nutcracker. They're not going to make the beta-galactosidase enzyme unless there is lactose around and there is no glucose around. And they are able to regulate all of this through a regulatory region called the lac promoter. And the lac promoter and the region of part of overlapping like lac operator, that's going to be this regulatory region of DNA that is in front of the genes for making beta-galactosidase, as well as a couple of other enzymes that the bacteria needs in order to use the lactose. And we don't really care about the use of lactose. We just want the machinery that is able to control when things get made. And so we take that machinery that controls that this beta-galactosidase and those other enzymes only get made in the presence of lactose and the absence of glucose, and we stick that machinery in front of our gene of interest. And now these bacteria, they're going to be like, oh, okay, so under these conditions, we're going to be repressed. If there's glucose around, if there's no lactose, we're not going to make the genes that are in front of us. But then if we make the conditions so that there is lactose around and there isn't glucose around, or if there is something that looks like the lactose product that is responsible for controlling things, then the bacteria will start making our protein, because our protein is in front of the instructions for making that machinery for the lactose production. And so basically we swap out all that lactose breakdown components and stick in our gene of interest, or the coding sequence for the protein that we want to make, basically. And then we have this regulatory information that the bacteria normally use to produce the lactose breakdown products, enzymes and things. And so now our gene is going to be under the control that the bacteria normally use in order to control the production of those lactose breakdowners. And so now we just need to manipulate the conditions so that the bacteria think there's lactose present and no glucose present at the point at which there's a lot of bacteria, and that way we can then control the bacteria switch to going from that mode when they're replicating to going to that mode where they're making lots and lots of our protein at the expense of replicating more. And so how do we get these conditions manipulated? The conventional way is with IPTG. And so IPTG is a mimic of lactose. It's actually a mimic of allolactose, which is the lactose products that actually, like, it's kind of like a version of lactose that if there's a lot of lactose around, some of it gets converted into allolactose, and that is actually the signal for bacteria that lactose is present. And what happens is that is part of that promoter. So the promoter overall, the promoter is basically where RNA polymerase binds. So RNA polymerase binds to the promoter in front of the gene. And what it does is it makes an RNA copy of the gene. So it makes a messenger RNA copy. That messenger RNA copy is used by the ribosomes in a process called translation to make the protein. So the production of the messenger RNA is transcription, and then the making of protein from that RNA is translation. And in bacteria, these are actually coupled, so as the gene is being transcribed, it's also being translated. And so you're able to really fast make a lot of protein. So the RNA promoter is in front of your gene, and part of that promoter is going to be, there's like an overlapping region called the operator. And on that operator is going to sit a Lac repressor, which is a protein that sits right there, and it blocks the RNA polymerase from coming and making things. And that Lac repressor is basically always going to be bound there unless allolactose binds to it. And when allolactose binds to that repressor, the repressor changes shape and falls off. And now RNA polymerase can bind and go and make the product. But RNA polymerase actually needs some help to get there. There's this protein called CAP, catabolite activator protein. And what it does is it actually helps bring the RNA polymerase to the site. If there's a lot of glucose, what's going to happen is that there's going to be a low level of cyclic AMP, CAMP, which is what kind of binds to CAP. Bottom line, high levels of glucose are going to prevent RNA polymerase from being recruited, whereas low levels of glucose, well, now you're going to actually get RNA polymerase brought to the site. And if you have lactose, well, now there's not a repressor protein bound there, and so RNA polymerase can bind and have a free ride to make transcription, translation, and now you're making your protein of interest. Just like the bacteria would normally be making that β-galactosidase and those other things that they need to break down the lactose. So we've tricked them into making our protein. And so we can trick them by adding IPTG, which will mimic that allolactose in order to cause the repressor protein to fall off. Or we can use autoinduction. And I like the autoinduction method because it allows me to be lazy. So with the IPTG induction, you need to make sure that you're timing things out so that the bacteria are at that point in their growth when there's lots and lots and lots of them. They haven't started overgrowing and they haven't started dying and things, but there's enough of them that you can get a lot of your protein made. And so what you normally do is you watch the OD600. So basically you look and see how foggy the media is, but you look with like a photometer. So you actually look in like a cuvette and things. So you're not just looking with your eye and saying, oh, that looks fuzzy. But you measure the OD600, so the optical density of 600 nanometer light, and you basically see when the bacteria are at an ideal point in their growth curve that you can then go and add the IPTG and say, okay, now switch over. But that means you've got to monitor it over time, and it's really easy to overshoot it. A lot more convenient is autoinduction. With autoinduction, however, here we don't have to watch. Instead, we just use smart ratios of sugars. And so we have this base media which has the sodium chloride, and it's got yeast extract, so basically just break open yeast and you've got a bunch of goodies in there. And it's got tryptone, so basically it's this protein digest. And so there's lots of goodies in there, but no sugar. But then we add a sugar solution, and so the sugar solution, it's got glucose and lactose as well as some glycerol. And it's such a ratio that the bacteria are going to start with the glucose because they like that better. They're going to grow. But before they start overgrowing, they're going to run out of glucose. And well, now it's time to switch over to lactose. And remember that when they switch over to lactose, they're not just switching over to make the machinery for breaking down the lactose, but they're also going to be making it so that they're going to make your protein of interest. And so they're going to start making the protein of interest that you want. And they're going to do this at this optimal point, and you didn't have to watch anything. So it is fabulous. When you make this autoinduction media, basically you make the base media and you autoclave it, and you then are going to add the sugar solution to it. You don't want to add the sugar solution before you autoclave it, because then you're just going to like caramelize things. But you still want to sterilize your sugar solution, so we vacuum filter ours. And so you just use a bottle top filter. I like to make some stock solutions of 8% lactose, 10% glucose, and 60% glycerol. And then we can actually pre-mix these together and then add that to our media. And then what we do is we have starter cultures that have been growing overnight. So smaller cultures and just normal LB media, normal media, and we then transfer some of that into the big flasks. Turn down the incubator temperature to 30. Let it go overnight, and we don't have to worry about anything. So there's some more complicated formulas for making auto-induction media, but we found this really simple way is just really easy. And so thank you for Walter Novak for helping, for telling us about this protocol. And this media is a lot easier than the ones that you see like originally published, but those originally published ones are more of a defined media. So if you want to have really good consistency, then that's where to go. So that's the basics of auto-induction. These bacteria really like glucose, and they'll only make this stuff for breaking down lactose if there's no glucose around and there is lactose around. So we, one, manipulate the conditions so that there's no glucose around and there is lactose around, and we, two, stick our gene for making our protein in front of the instructions for the control of all of that lactose breakdowning. And that way we're able to trick the bacteria into thinking there's no glucose and thinking there's lactose, and then being like, oh, okay, well, I'm going to go make the stuff for doing that. And then when they make the stuff for doing that, they're actually making our protein of interest. Psych! Typically what we do too is we actually have the T7 polymerase, the RNA polymerase that we want to make our protein for us. We have that under the control of the lac operon as well. And this allows us to have kind of like a dual layer of security where we have the T7 polymerase under the control of the lac promoter and our protein under the control of the lac promoter, and then needing the T7 polymerase. And so in that way, you're kind of able to really keep things from being expressed until you're ready. The bacterial strain BL21DE3, that DE3 part is for like this DE3 lysogen. Basically, the T7 RNA polymerase is from the T7 phage, which is a virus that infects bacteria. And basically the machinery for the RNA polymerase is in these strains of bacteria that are labeled DE3. And so those DE3 strains of bacteria will actually make the RNA polymerase, the T7 RNA polymerase, under these inducible conditions. And that's going to then allow you to make your protein under the T7 promoter under these inducible conditions. But then typically we also have the lac promoter with the T7, and then you can really have this multiple layers of security to prevent the bacteria from switching over and making your protein until it's time. And if you have some like protein that's toxic to the bacteria, there are additional layers of security that you can kind of add, because then it's really important that you don't have any leaky expression beforehand, where basically you get some expression, you get some protein made before you really want them to make the protein.